Hello and welcome back to Science of Medicine. In the first video on gastrointestinal tract, you saw how the anatomy and functionality of the gastrointestinal tract worked to take something that you put inside your mouth, extract nutrients from it, pass it through your gastrointestinal system. In this block, what I'd like to do is go through the details of what each section of your gastrointestinal tract is actually doing to accomplish those goals. So let's start with the oropharynx. So the oral cavity is made up of teeth in most cases, along with the tongue, and then several glands that secrete saliva. Now, the main function of this oral cavity is to macerate or mash up the big food particles, mix them with saliva so that you can put them in a bolus in the back of your throat and then swallow them, sending them down the esophagus. However, a few details on this. First off, that saliva also contains a little bit of amylase to begin digestion of carbohydrates. It's not that you need to chew your food to digest your carbohydrates. It's just to clean off the area in the oral pharynx. All of us know from personal experience that you can pass things through your nasopharynx that go into your oropharynx, and that's because in the back of your throat, in your oropharyngeal cavity, there is an opening to your nasopharyngeal cavity. And normally this is not what you want to do, but you do recognize that there is that ability to do it. Note also that you have several salivary glands which will begin secreting the saliva to coat this bolus of food that you're macerating in your mouth. When the appropriate coordination is needed, you will then take that bolus of food or liquid and put it into the posterior pharynx and then send it down your esophagus. Now, if you remember that the upper 1/3 of the esophagus is skeletal muscle, this is necessary for you to be able to control the initial bolus of food going down into the esophagus, and the lower 2/3 is smooth muscle and will pass that food or liquid bolus using peristalsis that we spoke about last time. Now remember that the trachea is in front, and the trachea has the epiglottis at the top of that portion. So when the bolus of food or liquid goes back, that epiglottis will go down and cover the opening that will go to your trachea, thereby hopefully ensuring that you don't choke and send stuff down the front. But all of us have an experience where we've had things that have aspirated into the top part of our respiratory system. And when you get it down to larynx, you can have that spasm when you get that liquid or a little bit of food that will actually be in contact with it to try to forcefully expel it, put it back into the esophagus and send it down the correct way. Now, once you've passed that food into the esophagus and you've had this peristalsis move it down the esophagus into the stomach, that will be your next place for this food bolus, for example. Now, the esophagus actually doesn't have a sphincter or a valve where it enters the stomach. In fact, it just enters through a portion of the diaphragm which has muscle around it that will create sort of a kind of sphincter that is more functional rather than an actual valve or constriction. So the esophagus enters the stomach at an oblique angle, and it goes through that diaphragm to allow for it to go into the portion of the stomach. We all have the experience where you can reflux things from the stomach back up into the esophagus. The most common thing that is done is the acid created by the parietal cells in the stomach, and that acid reflux is a very common complaint that people will have. Now, in the stomach, I will mix the bolus of food with digestive enzymes and acid, hydrochloric acid, which will be released from parietal cells that we'll talk about in detail in a minute. This will, through that muscular stomach, be moved back and forth to actually create a partially digested material which is called chyme. Now in the stomach itself, I have several types of cells that will be important for this process. The first are parietal cells, which I mentioned before, that are making and releasing hydrochloric acid. They also make and release a molecule called intrinsic factor. Now intrinsic factor is binding vitamin B12. Vitamin B12 is found in my red meats, and it will be digested by the digestive enzymes unless it's protected. And so we protect it with this intrinsic factor so that it doesn't get digested. We also have chief cells which secrete pepsinogen. We'll talk about this in just a minute. And then also, because we're going to digest proteins in the stomach or begin that digestion, and it's an acidic environment, we will auto-digest the parietal cells and the chief cells in the stomach unless we do something to protect them. So we have these mucus neck cells that will secrete mucus to sort of keep a little bit of a protective layer over the stomach cells. Now, they'll still get replenished on a fairly frequent basis. If I were to do endoscopy, that is to take a fiber optic tube and put it down the esophagus going into the stomach, I would see something like this picture. And this shows you this sort of glistening mucosa that is present in the stomach. If I were to take a biopsy of this, a piece of it, and then look at it under a light microscope, I would see something that is shown here. And let's use this to actually talk about the details of how the stomach is working. So up at the top portion of this is where the lumen of the stomach would be. And you can see these crypts going down with these cells that are lining it. Now these parietal cells that are here make and store hydrochloric acid. This is one of the most energy intensive processes in any of the cells in the body. And when the appropriate signal is obtained, for example, we talked about gastrin stimulating the secretion of hydrochloric acid, this will be released. And when food then is put down in the stomach, most of the time it's not going to have a pH which is really low, so it will actually have that HCl that will basically make it more acidic, but the food itself will titrate, if you will, some of that hydrochloric acid. So you release enough hydrochloric acid to try to lower the stomach contents to a pH of around 2. You may have heard of various antacids that are on the market. They work one of 3 ways. The first is something like Tums, where you would ingest it and its calcium carbonate. It would basically react directly with the stomach acid to neutralize it, or Mylanta, which would be a magnesium hydroxide. Another, which is the most popular type, are proton pump inhibitors. Proton pump inhibitors are molecules that will block the release directly of hydrochloric acid by these parietal cells, because they'll block the proton pump that is actually putting this HCl out into the stomach. There's another type which are called histamine type 2 blockers, and those are things like Zantac, and those actually also will ultimately block the hydrochloric acid secretion by the parietal cells, although the method by which they do that is less direct than the proton pump inhibitors. Now, the chief cells will secrete pepsinogen. Pepsinogen is an enzyme proenzyme. It itself is not the active form of the enzyme, but needs to be activated to form pepsin. And pepsin then, once it is present, is a proteolytic enzyme that breaks amide bonds in proteins. So it will begin the digestion of proteins when the pepsinogen at pH 2 is cleaved to be able to become activated pepsin. Now, the mucus cells are there to help from the autodigestion of this pepsin on the cells that line the stomach. Remember also I said that parietal cells release intrinsic factor that binds to the vitamin B12 to prevent it from autodegradation. So you eat a meal, it's gone down the esophagus and winds up in the stomach. Hydrochloric acid, intrinsic factor, pepsinogen are added to it, It's moved back and forth, and many of us have heard the rumblings of our stomach after we've eaten things sloshing about. And eventually I will produce chyme, this semi-solid material that will end up then being squirted out the end of the stomach through the pyloric sphincter. And it will then start to go into the small intestine. So the first part of the small intestine is the duodenum. And a lot of things go on in the duodenum, so we're going to have to look at this very much in detail. So I've squirted my chyme out through the duodenum, and it goes into a very complicated type of structure. So a lot of things are happening in the duodenum. The first thing that I want to show you is some anatomy, some detailed anatomy. The duodenum is like a C-type structure, so it forms this C-type structure, and a number of things dump into the duodenum. At the proximal end of the duodenum, I have chyme being squirted out from the stomach, and so it's this pH2 stuff that is actually present with the pepsin. I also have a few ducts coming down. I have a duct coming from the liver and also from the gallbladder that is going to release bile and other things that come from the liver that we'll talk about in a few minutes. And that's coming down from the liver. I also have the head of the pancreas nestled within this C shape of the duodenum. And that exocrine portion of the pancreas has a ductal system that will release also through a little sphincter called the sphincter of Oddi that will release various things. In particular, the first thing it will release, or one of the things that it will release, is bicarbonate ions to neutralize the pH or get it fairly close to a neutral pH. And that's because the enzymes that come from the pancreas have an optimal pH of between about 6 and a 7. So I'll have trypsin, chymotrypsin, carboxypeptidase, which will break down further the proteins into their component amino acids. I'll also secrete pancreatic lipase, which will help start the digestion of lipids, and also amylase that will help with the digestion of carbohydrates. Now, the bile that is coming from my pancreas is also necessary to help emulsify the fats so that I can then assist with the absorption of things like fatty acids, lipids, and cholesterol. So a lot is going on with what gets secreted or squirted out of my pyloric sphincter into my duodenum. I do have the absorption of some things. For example, vitamins that are fat soluble, A, D, E, and K, are absorbed in the duodenum. Now, I know you remember from when we talked about skin that I can also synthesize vitamin D in the skin, and that's absolutely true. But I can also absorb it in the duodenum. I also will absorb calcium and iron. And the reason that I mention this is because one of the things that is fairly prevalent in the human population is a disease called celiac disease. And celiac disease is in the duodenum and leads to lower absorption. So a lot of times people with celiac disease will actually have deficiencies of these nutrients that are actually absorbed in the duodenum. So I've now gone through the duodenum, and I'm going to pass into the remainder of the small intestine. The first part that I will pass into is the duodenum, and then afterwards is the ileum. And this is very, very large, many, many meters of digestive and absorptive capabilities. And not only that, the absorptive surface is not only because I have this undulating tubular structure that's going back and forth in my gut, but I also have projections within that are villous projections, like finger-like projections in there. And then in addition to that, I have microvilli that increase the surface area there also. Now, the duodenum and the ileum is where I'm going to actually have the final digestion and absorption of products of my digestive system. I'm going to absorb small ions, I'm going to absorb all kinds of small molecules, water is also being absorbed in there. Now, these cells actually will have a very short lifetime in my gastrointestinal tract, generally just a few days. And this is going to become important, and I'll talk about that in just a second. If I look at a light micrograph of the jejunum, or the small intestine, or the ileum, I find these crypts going down here. And I have several different cells that line these projective villi, and then the villi having little microvilli that are on a microscopic scale here. So if I draw just sort of a pictorial representation of this, what I find is that these are the cells that are lining these microvilli. They start from the bottom and they just form their way up. They have a lifetime of about 2 to 3 days, about 3 or so days, and so they're shed constantly as they make their way up to the top. But underneath them, I have a highly vascular system. All right, and that vascular system is going to go into my portal vein, which is then going to transport my nutrients that are absorbed into the liver. I also have a way to absorb the fats through what is called a lacteal, to be able to emulsify those and bring them in. So what's happening here? So carbohydrates, proteins, and fats are what I'm going to actually bring into the body in some form. The carbohydrates have been digested in large part with the amylase, and this breaks them into small pieces of sugars. And there are separate enzymes on the microvillus cells that actually will finish off digestion. So I have things like lactase, sucrase, maltase, dextrinase, all of those small polymers or pieces of sugars where you put together 5 carbon and 6 carbon, or 2 6-carbon sugars, and you want to break those apart. And that's all on this brush border of this microvilli, and then you will bring in those sugars. Now, fortunately in the body, as I mentioned before, that even though glucose is what's going into the Krebs cycle, if I have 5 carbon sugars, I can convert those into things like glucose. I can take 1 glucose and put it into another form that I can go into the Krebs cycle. So I have the capabilities biochemically to be able to do that. Proteins, I've digested a lot with the pepsin, and then also the trypsin, chymotrypsin, carboxypeptase, that cleave those polypeptides at various places. I also have these types of enzymes that are along the brush border to break down the proteins into their component amino acids, which I can then bring in. And as I mentioned before, the fats, I will have lipase to help digest my triglycerides, and then I can emulsify other fats with things like bile or lecithin, and then bring those into the lacteal. So once I've passed through the small intestine, I will then go through the ileocecal valve, which is located just before the ascending colon, and pass those constituents that have not been absorbed into the colon. The colon is comprised of the ascending, or going up, transverse going across, and descending colon, until I get down into the sigmoid area, and then eventually into the rectum and out the anus. Now, in the colon, I have a few important components still left to extract from my material. The first thing is I'll take out some water and electrolytes that were not absorbed in the small intestine. But remember also that the stomach parietal cells released intrinsic factor, and intrinsic factor bound to vitamin B12. This will then come off as I go into the colon, and that will allow for vitamin B12 to be absorbed in the colon. Whatever passes through the colon and goes down into the sigmoid colon and into the rectum will now become my fecal matter. That'll be my waste. And I'll store that in the rectum until I get an appropriate signal that says that I want to go ahead and move this out. And then I have a little sling of muscle that I can control to allow the rectum to allow for the release of fecal matter through my anus. Now, there are some accessory organs that I've mentioned. I talked a bit about the pancreas, I want to talk a little bit about the liver now. Remember that the liver was sending through a ductal system material into the duodenum, and then also I have the gallbladder here. And there are a lot of things that go on in this area, as you can see with this picture. Remember, I have blood coming from the gastrointestinal tract through the portal vein, and then I also have a hepatic artery, which comes off of the celiac artery that's taking oxygenated blood and going through my liver. Now, my liver is getting all of these nutrients. It's getting oxygenated blood. It's getting the nutrient-rich blood, and it has a lot of things to do. So I break these up into things, the major things that it's doing. First off, the liver is involved with making things. It's involved with degrading things, and it's involved with storing things. There are many, many more things that the liver does within these domains, but these are very important and things that are touched upon in science of medicine, and specifically in medical physiology. So on the synthetic side, there are proteins that are made in the liver. There are the coagulation proteins that are necessary for you to clot, complement proteins necessary for the innate immune system, and albumin, which is one of the main determinants of the oncotic pressure within your blood. We have carbohydrate metabolism and the ability to make glucose that can feed into the Krebs cycle from other things other than just simply having glucose. So that process is called gluconeogenesis. I make cholesterol in the liver. In fact, much of the cholesterol that you have in your body is not what you ingest, but in fact what you make in the liver. I also make lipids and bile. Bile, remember, being that emulsifying agent that is going to help with it, much like a detergent in emulsifying my lipids. And then I make hormones. In particular, I want you to remember that insulin-like growth factor, which is one of the things that we will talk about in the endocrinology section, is made in the liver, as well as thrombopoietin, the hormone that stimulates the production of platelets. I degrade things in the liver. I degrade things like hormones, in particular insulin. And then I also have drugs and toxins that I ingest. If I take some acetaminophen or some ibuprofen, that's something that my body is not normally recognizing as food, because it is not, and so it considers that a toxin and tries to get rid of it by making it more soluble or by making it such that it can go out through the gastrointestinal tract. And then the urea cycle, where I take ammonia that's produced from proteins and send that into a cycle that makes urea, which is less toxic, those enzymes are present in the liver. I store things in the liver, and amongst one of the many things that I store is glycogen, which is a polymer of glucose that allows me to break down very, very rapidly and then produce glucose and make that in my bloodstream very, very quickly. I also will store vitamin A, B12, and K in the liver, as well as copper. And the reason that I mention copper is that there's a disease called Wilson's disease, which has to do with a storage issue with copper that is present sometimes. It's a fairly rare disease, but can cause an issue within the liver. Now, how does it do this? So there's some structure that's important here. First off, the liver is taking the blood from the portal vein, and that blood is going into a little triad where I have an arteriole, which is coming off of the hepatic artery, this portal vein here, and then this bile duct, which is going down and creating things. And I have these sinusoidal cellular areas where I have the hepatocytes, or the liver cells, and they're mixing and meandering down through until they get to what's called the central vein, which eventually then ends up forming a large vein, which then goes back to the right atrium of the heart. And if I look at the detail of this, what I find is that on my triad over here, I'm having my portal vein flow coming here, I'm having my hepatic artery vein flow coming here, and going past these hepatocytes. The hepatocytes are very busy synthesizing things, degrading things, they're doing all of this. Some of the waste is then going down into this bile. They're also making bile, but they're sending it down through that bile duct, and that can get dumped into the duodenum, as we saw earlier. Eventually, then things that are made or degraded then will go through that central vein and will wind up going back to the heart. Very important, and lots of things that are going on. The transport of blood through here is very slow. It's not a very fast process like it would be through an artery. It's meandering to permit time for the cells in the liver to do their thing. Now, structure is really, really important to the function. And if you look at hepatocytes that are normal, you'll see these kinds of sinusoids in this. But in a person that's had damage to their liver from, let's say, cirrhosis, you get this fibrous material that surrounds these structures, and it inhibits the functionality of the liver. And that's why liver cirrhosis, either through things like alcohol consumption or through fatty liver, or what we call non-alcoholic steatohepatitis, can lead to a breakdown of the structure of the liver and therefore its functionality. So let's talk about some questions. So let's say a patient has a Billroth gastric bypass surgery. And in this process, you remove a sizable portion of the patient's stomach, and it's directly connected with the first part of the jejunum. Which of the following might occur and why? So I'll have you look at these, think about them for a moment. You need to pause the video to think about them. That's great. All right, now I hope that you recognize that the problem that you might have would be vitamin B12 deficiency because of having less intrinsic factor, because you've taken off a portion of the parietal cells by bypassing part of the stomach and going directly into jejunum. You would not have the B12 deficiency from less absorption because that's occurring in the colon. Amylase doesn't have anything to do with protein digestion. Carbohydrates digestion is not done in an acidic environment. The amylase is present at it's active at a higher pH. And vitamin D deficiency doesn't have anything to do with bicarbonate per se, and you can also synthesize that in the skin. Let's try another one. Let's say you have a patient with end-stage liver disease. Which of the following would you expect that this would happen? I'll give you a chance to read those. Now, I hope that you would recognize that you would likely have lower extremity edema in both legs or in any kind of dependent area. And this is because you have less albumin synthesized when you have end-stage liver disease. Remember, that was one of the functionalities. Now, excessive blood clots would not be the case because if you remember, you make the coagulation proteins as well as thrombopoietin in the liver. So both of those are involved in clotting. If you don't make them, you're going to have problems with clots, not excessive clots. And remember, you break down insulin. So increased blood glucose levels would not be the case because insulin is involved in actually bringing insulin into the body. If you've got an excess around it, you're not going to have increased insulin levels, increased glucose levels. Absorption of iron is occurring within the duodenum and not in the liver. And high fat meal and enhanced ability would not be the case because you're making bile and other things to emulsify those fats in the liver. If you're not making them because you have end-stage liver disease, you're not going to have an enhancement in that ability. So I hope this has given you an overview of the gastrointestinal system with some of the details. As always, if you have any comments, please leave them and feel free to subscribe so that you know when new videos are actually released. I look forward to seeing you next time on Science of Medicine.